Hello everyone. My name is Debbie, and for the Honors Independent Research course this year, I conducted a reverse genetic research on a marine microalgal species called Nanocoropsis oceanica in order to try increase its thermal maximum, thereby increase the maximum temperature at which they can survive at. Okay, so from a broader scheme, I want to start by noting the importance of marine microalgal species to the aquatic ecosystem. The reason why they're so essential in their environment is because they make up the base of the food web in the aquatic ecosystem. It's been shown that they are responsible for around 50% of the primary productivity as well as the oxygen production, which means that they make up a significant portion of the total energy and the oxygen that is produced in the ocean that is needed by other organisms higher up in the food chain to survive. In addition to this, they are vital to help mitigate climate change as they have a much higher efficiency rate at fixating carbon dioxide compared to terrestrial plants. Now, due to their vast importance, it becomes important for us to note how the increasing marine temperatures due to global warming can affect marine microalgal species. Now, this can lead to two contrasting effects. On the top is an increase in the growth rate of these algae, and on the bottom is a decrease in the growth rate. And this contrasting phenomena is mainly attributed to the thermal maximum or the thermal tolerance that is unique to each species of algae. The thermal maximum is the maximum temperature at which organisms can survive in. And because this is different for each species of algae, those that possess a higher thermal maximum are able to see a greater growth rate at these elevated temperatures. However, if the surrounding temperature surpasses the thermal maximum of certain species, it will disrupt their biological processes, which can lead to a decrease in their growth rate and further to their mortality. Now, then it might become obvious that there are several problems that can arise in the aquatic ecosystem due to a decreased growth rate in several species of marine microalgae. First being a loss in biodiversity. With less algal species available, there's less oxygen, energy, and nutrients available for other organisms as well. And a more specific issue, and the issue that I personally wanted to focus on for my research, was coral bleaching. And this is something that can occur when Symbiodinium, which are marine algae that live in coral reefs, start to die off because they cannot withstand the elevated temperatures. And when this happens, because the Symbiodinium are responsible for around 90% of the total energy supplied to coral reefs, the reefs will also become vulnerable to starvation and diseases and will experience bleaching. And prolonged coral bleaching can lead to their mortality as well. Now, then it becomes clear to us that there's this need to conserve marine microalgal species against the continually increasing temperatures. However, the problem is that there's a lack of genetic understanding in the specific genes that affect the thermal tolerance of marine microalgal species. And there has been past research that aimed to identify these genes and also to try increase the thermal maximum of these organisms, some of which included sequencing already existing clades that exhibit this phenotype, or prolonged exposure to elevated temperatures, or even a CRISPR-Cas9 based genome editing. However, due to the limitations that are attached to many of these research, there hasn't been yet an identification of a very specific gene or genes that can be mutated in a way to increase the thermal maximum of these organisms. Hence, for my research, I rose the question, what are the specific genes that can be mutated in marine microalgal species to increase their thermal maximum? And I split this question into two different steps. First, to produce a strain of marine microalgae exhibiting a higher thermal tolerance, and second, to identify the specific genes or mutations that allow for this phenotype. Now, I mentioned earlier that I want to focus on the issue of coral bleaching, and to do this, I initially want to work with the Symbiodinium algae. However, due to their genomes that are much bigger and complex, I decided to use another species called Nanocoropsis oceanica as a model species to gain a better genetic understanding of the organisms as a whole. And the Nanocoropsis were a great candidate because they had genomes that were fully annotated and assembled, and it was also relatively smaller. They had a faster growth rate, and most importantly, it's been often used in past research. So I had a much more extended amount of genetic resources that were available to me by using the Nanocoropsis. As for my methods, I first prepared and performed maintenance tests on my algal culture. I kept two different medias, the solid agar plate and a liquid culture. And this is because it was easier to observe their growth on the solid plates. And I needed the liquid culture for a later step called the electroporation. After preparing my algal cell culture, I performed the controlled thermal trials. And the purpose of this was to establish the thermal maximum of the nanocropsis before transformation to compare it to the algal cells after transformation. And for my step number 3, alongside step 2, I searched for the promoter region. Promoters are short gene sequences that basically start transcription. And transcription is a process where DNA gets converted to RNA, which through a series of other steps can code for proteins, which can allow organisms to exhibit different traits. However, each promoters have different transcription efficiencies. And because of this, I had to search in the literature to see which promoters had high transcription levels, specifically in Nanocropsis oceanica. And from this search, I found that the RIBI and the HSP70 promoters would be suitable for use. And from here, I had to design the primers myself to perform a step called PCR, which allowed me to make millions of copies of the same promoter. After these preparation steps, I performed the electroporation, and this was a technique I used to cause the mutations in the algal cells. So electroporation is a technique that basically utilizes very high electrical field strengths in order to open up small pores in the cell membranes of the algae. And through these pores, I was able to insert the promoters I've made earlier in step number 3 into them. And the overall rationale I followed for my methods was explained by random mutagenesis, which states that if I insert millions of promoters that I've made into the algal cells, they'll attach to different random regions in the genome, and they'll start transcribing these regions, which will lead to millions of mutations. And it just had to be one of these millions of mutations that allowed the nanocropsis to exhibit a higher thermal tolerance. To search for the specific colonies that were able to uptake the specific mutations we needed, I conducted a heat screening trial. And this was basically where I placed all the transferrin colonies into incubators at the thermal maximum temperature established earlier in step number 2. And this is a temperature where they're supposed to die off in. So if there were any mutated colonies that were able to survive and grow at this thermal maximum temperature, it would indicate that these are candidate colonies that potentially were able to uptake a mutation that allowed them to exhibit a higher thermal tolerance. So then I took these candidate colonies and sequenced them in order to identify exactly where the mutations occurred and what types of mutations occurred for a better genetic insight. Now, for my results of the controlled thermal trial, I was first able to establish the thermal maximum at 33 degrees Celsius because this was a temperature they didn't grow at or didn't recover at after 32 days of trying to observe growth. And for my electroporation, I was able to confirm the viability of the protocol that I was using through a plasmid transformation. So this was not explained earlier in the methods, but in this step, I essentially wanted to make sure that the electroporation protocol itself that I was using was successful and working. And this is so that I can know that when I'm trying to insert the promoters through the electroporation protocol I'm using, I want to make sure that the cells are actually uptaking these promoters and causing mutations. So to confirm this procedure, then, I inserted plasmids that had antibiotic-resistant genes in them and inserted them into the nanocropsis cells using the electroporation protocol. And as you can see in figure 2, when I plated them in antibiotic plates, in the control, there is no growth of any colonies. And this is good because algal cells aren't supposed to just have antibiotic-resistant qualities. However, in plasmid 1, 2, and 3, you can see that there's growth of colonies, which means that the cells were able to uptake the plasmid, the genes that were in the plasmid, meaning that the electroporation protocol I was using was viable and working. So from here, my heat screening results, I was able to observe potential colonies that were exhibiting a higher thermal tolerance through the promoter transformation results. In figure 3 shown is my round 1 of electroporation. And in my first round, there were no colonies seen growing at the thermal maximum temperature. However, in figure 4, you can see on the bottom are 3 tiny little colonies that were growing at the 33 degrees Celsius, at the thermal maximum, which is where they're supposed to die off in. And this was very exciting news for me because it means that I was able to cause some type of mutations to these specific colonies that allowed them to grow at temperatures that they're supposed to die off in. And out of these 3 colonies, the one pictured above RIBI number 3 was the only one that survived after the 10 days. And I'm currently in the process of sequencing this colony right now to identify the locations of the mutations. Now then, all in all, Um, the overall finding of my research was that I produced a potentially heat-tolerant colony of the Nanocoropsis. And there's several implications to this, two of the main ones being that it's a proof of concept. In the literature, there is a minimal number of research that combines the use of electroporation and random mutagenesis in order to produce a heat-tolerant strain of marine microalgae. However, the fact that I was able to create and produce this potential colony with this desired phenotype shows to other researchers that this is a method that can be used to produce different phenotypes. And alongside this, my finding is in addition to the genetic understanding of marine microalgal species as a whole, especially in the realm of their thermal tolerance, which there's a lack of genetic understanding in currently. Now then, further research will expand the findings that I was able to find from this research. As I mentioned above, sequencing the potential colony I found will allow me to identify specific gene regions. And from this, I would like to use different genetic engineering techniques, such as the CRISPR-Cas9 based editing, in order to target the specific genes I've identified in order to further confirm, do these genes really allow the nanocropsis to exhibit a higher thermal tolerance. And from here, I hope that I can apply the baseline understanding from this research to the symbiodinium algae, which have genomes that are much bigger and harder to work with. Um, to end my research, a reflection for future students, do not procrastinate because many of you in this room know that I'm the biggest procrastinator and it will not cut it for this course. This is not a course where you can just cram for 1 or 2 weeks to achieve the results you want. You really have to put in a steady and consistent effort for several weeks and months in order to achieve the results you'd want. And alongside this, have a good and specific lab notebook. There were a lot of protocols within this research, PCR, DNA extraction, sequencing, that were quite complicated, and having a detailed notebook to look back to really helped me in the future. And big mahalo to Dr. Chan for helping me throughout my entire research journey, my mentor Panza, as well as my other teachers, Mrs. Kobayashi, Dr. Perez David, and Mr. Hill for guiding me through this journey. Thank you.